Hello, everyone. Welcome to the Problem Set 10 worksheet video. So today our topics are biochemical pathways and mutation. We'll spend most of our time on the mutation part, but let's start off with one biochemical pathway problem. So as you can see, this is a branch pathway, and the normal strains can produce this end product. All of the mutant strains cannot produce their end product. They're defective at some step or another along the line. So you notice I've got metabolites between the arrows because those are the intermediates and precursors and so forth on the pathway. And then we'll put strain names over the arrow. The strain is always placed over the arrow at which that strain is defective. So in this case, strain number 1 cannot convert metabolite C to metabolite E. Our first question is which metabolite or metabolites will restore the normal phenotype for strain 1? The rule is if this step is blocked, everything on the same branch all the way down to the end can restore that wild type phenotype. So we could supply metabolite E or metabolite F and get the normal phenotype produced. So let's look at it the other way. Suppose strain 2 can be rescued or make the end product when we incubate with metabolites B, D, or F, but it can't be rescued with the others. So B is here, D is there, and F is here. So strain 2 must be defective in this enzymatic step because that way metabolites B, D, and F would all come after the place where strain 2 can't make the enzyme. I hope that's helpful. So the rest of our problem session is going to be devoted to mutations. One thing that it's handy to have in front of you is a coding dictionary. I encourage you to print one off and keep it when you're doing your homework problems and your practice problems. But of course, you can't use that on the exam. However, on the exam, the very first item on the exam is going to be a coding dictionary. So let's take a look at problem 2, and we'll refer to the coding dictionary when we need to. A codon that specifies the amino acid glycine undergoes a single base pair substitution, resulting in a nonsense mutant. So we want to classify this mutation. Is it a point or a frame shift? Well, our clue here is single base pair substitution. That makes this a point mutation. Remember, a frame shift mutation inserts or removes a base pair from the DNA, whereas a point mutation substitutes one base pair for another. Next question is, what is a nonsense mutation? A nonsense mutation is a mutation that starts off with a codon, or the coding sequence starts with a codon, but the mutation causes that codon to be changed to a stop codon. And it's also typically going to cause an early termination of the protein. So it will make a shorter than normal protein. We can look a little bit at our coding dictionary. We see that we started off with a codon specifying glycine. So glycine is G anything, and the stop codons are UAA, UAG, UAG, and UGA. So the idea is this question is going from a codon like this, where that blank can be anything, to one of these 3 codons. Well, we've got a couple more questions to answer before we actually solve that part of the question. What is a transition? A transition is a point mutation where we're going from purine to purine or from pyrimidine to pyrimidine. So something that went from a guanine, which would be a GC base pair, going to an adenine. The guanine changed to the adenine, which means we went to an AT pair. That would be a transition. Or we could have, and that's purine to purine, we could also have pyrimidine to pyrimidine. A transversion is going from one type of molecule to the other. So if we started off with a GC base pair, going to something that was a TA, so the guanine changed to the pyrimidine thymine, and the cytosine changed to the purine adenine. So was the mutation from glycine to the nonsense mutant a transition or a transversion? Now we need to look at these codons a little bit more carefully. GG anything codes for glycine. We want to change only one of these three letters and get to this last stop codon. If I go from GG anything to UAA, I have to change two. Similarly, if I go from GG anything to UAG, I have to change two. But if I go from GG anything, and that anything happened to be an A, and I went to UGA, I only have to change that one position. So this must be my glycine codon, and that must be the stop codon that I used. That means the change that I had was an original GC base pair right at this first position of the codon going to a TA base pair. Because remember, on the DNA basis, mutations occur at the DNA level, and at the DNA basis, that uracil would be a thymine. So that is going to be a transversion. So I've got a couple of other questions to ask about some of the vocabulary. Another word for a silent mutation is a synonymous. That means that the codon is changed, but it still codes for the same amino acid. And another word for a missense mutation is a non-synonymous. And that means the codon was changed to another codon that coded for a different amino acid. Okay. Well, now this one looks like it's got a little bit more work to it. We've got a mutation that alters by either adding or deleting one base pair, or changing, like causes an alteration, an addition, or a deletion. So we can have a point mutation or a frameshift mutation to get from the normal protein sequence to the mutant sequence. And you want to figure out the RNA sequences, the messenger RNA sequences for each of these proteins to the best of your ability. And it may not be a perfect thing. And then we will be able to figure out where the mutation was, figure out if it was point or frameshift. And then, if it was a point mutation, was it a transition or a transversion? If it was a frameshift, was it an insertion or a deletion? And maybe we can even figure out the stop codon that was used in the shorter protein, which happens to be the mutant protein. So the way I start this is I write these amino acid sequences out and I space them. So I've written the normal sequence here and the mutant sequence there. And then I use my coding dictionary and I figure out as much as I can about the codons used. So I prepared this before I started the video, but let me walk you through the first little bit. So methionine, AUG, codes for methionine. I write AUG down. Histidine, CA, either U or C. So I've written CA, and then I've written a line with U and C above it, and I'll try to figure out which one it is as I go along. Let's see, here we go. GA, either U or C. For the next one, glycine is, whoops, that should be a GG, either anything. And then tryptophan is only one thing. Tryptophan is UGG. I've done the same thing with the mutant, AUG for methionine. For glutamine, CA anything. And where is glutamine? CA, either A or G, not anything. And now I've done one other thing. I put the stop here. So I actually wrote one stop there, but what I need to do is write the word stop here and then write all three stop codons, UGA, UAG, and UAA. Because if this protein stopped early, it stopped because somehow there was a stop codon there. And this is something we can often figure out with the shorter protein. We won't be able to figure out what stop codon was used on the longer protein. So then it asks, I'm looking at the questions here, was the mutation a point mutation or a frame shift? Now, just by looking at the sequence of this protein, we can actually figure that out. By looking at this protein sequence, this one stopped early and there was a difference in the second amino acid here. So that's two things going wrong, which means this had to be a frame shift mutation. Now we need to figure out, was it an insertion or a deletion? This is where I compare these two DNA sequences. AUG, that's the same. CA, here's a difference. And then the next thing I need is a stop codon. If I looked and used that U and that GA, I would have this stop codon, which means whatever was added in this position was added. And we can't tell if we added adenine or guanine, but we know we added something there. So that will give us the idea of this was an insertion and we put something in at this particular point. Let's see. The next question, which stop codon was used? Well, we figured that out, UGA. So that was our stop codon. And then I think we answered that question. So it was an insertion. We inserted an AT pair would work, putting an extra adenine in here. It could have been the guanine as well. Okay, next question. Again, I've got a coding strand of a normal sequence here and a mutant sequence here, and ask if this is a point or a frameshift mutation. Well, these don't line up perfectly, but they line up pretty closely. They look to be the same length. So I'm suspecting that this will be a point mutation because one's not longer and one's not shorter. So let's compare these sequences and see if we can find something that differs between them. Ah, here we go. Right there. So this is a point mutation. And it appears that I went from a CG base pair to a TA base pair because I went from C to T. So that's going to be a transition. And it says, give the one-letter abbreviations for the amino acids in the normal protein sequence. So we're going to translate that normal protein sequence. And Moodle, I actually did this similarly to one of the questions in your homework. Moodle is very particular, as we all know, about your syntax when you type in answers. And this asks you to not put any spaces between the letters so that Moodle will grade it right and write it from the amino to the carboxyl end. So our normal, we start at the 5' end, find our first start codon, ATG, and then break things into codons. And there's a stop. TGA is like UGA. ATG codes for methionine. So the first letter is M. GCC. GCC codes for alanine. So that's going to be an A. AAU codes for asparagine, that's an N. GAC codes for aspartic acid, so that's D. And what was my last one? UAC. UAC codes for tyrosine, and that is a Y. So our protein actually spelled a word, Mandy, which was one of the names of my flat-coated retrievers. So sometimes these proteins will spell a word, sometimes they won't. So if it doesn't spell a word, don't panic. They don't always spell a word. You would expect when you're translating, though, that your first letter is going to be M, because your start codon is AUG. On the DNA level, it looks like ATG, and that codes for methionine, abbreviated M. And then give the one-letter abbreviations for the amino acids in the mutant sequence. Okay, that's going to be the same. That's going to be the same. That's going to be the same. That one's different. And then these two, that's the same, and that's still a stop. So we know it's going to be MAN. And then GAU. Go to my coding dictionary, GAU. GAU. Ha, 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 look at that. GAC, GAU. Still codes for the same thing, doesn't it? It's still a D because of the degeneracy of the code. Both of those codons happen to code for the same amino acid. And then, of course, UAC will still be Y. So in this case, we would say this is a synonymous mutation. And another word for that is silent mutation. Okay. Let's try another one. Another piece of DNA here. And again, although things don't line up perfectly, it does appear that we can tell in this case a difference between the lengths of these two DNA sequences. Looks like the second one is a little bit longer. So I'm guessing this is a frameshift mutation. And we'll go along and see if we can figure out where the change is. G, 3 A's, T, G, A, C, T. We have 3 A's here. We only have 2 A's there. We don't know which adenine was added, but there's an insertion of an AT pair. And I'm just going to circle the third one, but it would have been equally correct to circle the first or the second. So this is a frameshift. It is the insertion of an AT pair in the DNA. And like I said, it could have been any of those three. We can't make a distinguishing change there. And then it says to give the one-letter abbreviation for the amino acids in the normal sequence, and then you're going to do the same in the mutant sequence and figure out how to classify this particular type of mutation. So the normal, start off, go across from the 5' end. We read our ATG as AUG. I'm just going to divide this into codons till I get to a stop codon. And let's see, AUG codes for methionine. ACT, it's like ACU, codes for threonine. AAU codes for asparagine. And GGC codes for glycine. MT and G. Didn't spell a thing this time, did it? Now let's look at the mutant. We'll just split it again into codons until we come up with a stop codon. Here we go, way at the end, UAA. So M, ACT, still going to be the same thing. AAA, it's going to be lysine, it's a K. UGG is tryptophan, that's a W. CUG, there it is, leucine, it's an L. ACA, threonine, that's a T. GCA is alanine, that's an A. So I'm going to write this down here. Let's see, the normal one was MTNG, because I know that's kind of hard to read. And then I'm writing these letters for the mutant one down here, MTKWLTA. This is going to be classified as a read-through mutation. When the protein doesn't stop on the right time and goes longer, we have a read-through. When the protein stops early, we have a nonsense. And then silent and missense have to do with the synonymous and non-synonymous mutations. Let's take a look at another. A gene encodes a protein with the following amino acid sequence. And then there's a nonsense mutant, stopping early, that is a single base pair substitution. So nonsense mutants can occur with a frameshift mutation, but they can occur with a substitution, a base substitution as well. If, like in that first problem, we change the codon from one coding for an amino acid to a stop codon. So the short mutant protein only goes met-lyse. And then we can think about that as having a stop codon there. Then we have an intragenic suppressor. That means within the gene, there's a suppressor mutation that gets it back on track. It doesn't make the perfect protein sequence. In fact, this is the protein sequence, and you can see it got messed up for a base or for an amino acid, and then it got back on track. And so this is going to restore the reading frame. So if we changed one, and you notice we didn't change it perfectly. We changed it, this changed the serine to a stop codon, and then this changed the stop to a cysteine. So it didn't change it back to serine. But sometimes two wrongs don't necessarily make a right, but they get it back on track. So we want to give the location and nature of the original protein mutation and of the suppressor mutation. So the original coding for serine. Let's look at all of our serine codings. There's a whole bunch of them. You see anything, and then there are a couple over here, A, G, either U or C. So all those code for serine. Let's look at our stop codons, because we know that the codon for serine was changed to a stop codon here, and those are UAA, UAG, and UGA. There we go. And then we need to look at our cysteine codons. And cysteine is over here, UG, either U or C. So the question is, how did we get from one of these codons to one of those codons, only changing one base, to one of these codons? So if the original codon was UCA, so I'm just going to scribble it down here. Say my original one was UCA for serine. And I'm doing that because I have a UGA stop codon. So that A means that this blank would have to be an A. And the U did not change, and the C changed to a G. And then this had to change UG something. So this had to change to UG, either U or C. So going from this first one to the second, we change this position, and that's going to be a transversion. Going from this position here to that, no matter how you look at it, it is a transversion because this is an AT pair and we're going from a purine to a pyrimidine. So that is also a transversion. Whoops, there we go. Now you can see it a bit. So I did a transversion from a CG base pair to a GC, and then another transversion from an AT base pair to either a TA or a CG. I encourage you to practice these. They start coming a lot easier as you practice them. Let's see, I think there's one more page of these. There is one more page. This one's kind of long. We've got a normal sequence of the coding strand and a whole bunch of mutant sequences. And the ideas are saying that every change that does change the protein sequence changes the function. So we might have a change that doesn't change the protein sequence, but if it does, it will change the function. And you want to classify everything as a transition, transversion, frame shift insertion, or frame shift deletion, and then describe what effect that will have on the resulting protein. So I'm going to do the first one, and then I'm going to let you do the others on your own and check yourself with the key. And of course, you can email me if you have any questions about it. Well, let's look at these first one too. I'm comparing the normal to mutant A. Looks like TGA. Whoa, here's a difference right here. And everything else is the same. So I went, the mutation is going from C to G, which means I went from a CG base pair on double-stranded DNA to a GC, and that's going to be a transversion. Let's just take a quick look and see what that would do to the protein. To do that, we have to start with our start codons right here, and then we'll be translating using 3-letter codons. Those are our stops. These don't change at all. Those don't change. The only base we changed was right here. So that means the only codon we messed up was right here. ACU, and I'm just going to scribble down here. Our codon of ACU went to a codon of AGU. Now let's bring in the coding dictionary and see what that means. ACU coded for threonine, and AGU, codes for serine. So that is a non-synonymous mutation or a missense. I hope that this has been helpful for you. I encourage you to work as many practice problems as you can, and I encourage you to try them closed book. This will help you identify areas where you're struggling, and you can come back and pay a little bit more attention in your studying to those areas. Please do contact me if you have any questions. Thank you for your attention. Have a great day.